Hello, this is lecture 26, the last lecture in social neuroscience, in which I'm going to talk about the future of social neuroscience, but I'm also going to talk a little bit about the past and the present in social neuroscience. So in sort of wrapping up all of these many lectures, we're going to talk about some of the general limitations and strengths of social neuroscience today. I'm then going to talk about some things that I think are going to be found in the next wave of social neuroscience. And finally, I'll end just with the thought about, let's say you're considering a career in social neuroscience. What would be the next steps? All right, let's start with part one, limitations and strengths today. So the limitations of social neuroscience, I have to say that even though it's been around 30 years or so, it's still in its infancy. There still needs to be a lot more done in social neuroscience, and I think that's actually kind of exciting. But it's also an expensive research endeavor. As you can imagine, many of the methods we've been talking about cost money. They cost, you need to get grants to pay for the equipment. Even if you're doing things like hormones, you're going to have your cortisol and testosterone assayed from saliva samples. There's a lot of money here. So neuroimaging costs a lot of money. It's still very expensive. It's something I can't do at the moment because I don't have a particular grant to pay for my own neuroimaging. But blood chemistry costs money. If you want to do neuropsychology and you're trying to find patients that have certain brain damage or lesions, that's expensive to recruit them and find them and get them scanned and so on. So it's just an expensive research endeavor. That's something to keep in mind as a limitation of social neuroscience. Another issue about social neuroscience is its ecological validity of a lot of its experimental paradigms. As you've seen, a lot of these studies, especially the studies that involve fMRI scanning, have been studies that seem quite artificial, trying to get somebody to feel provoked, for instance, or feeling angry while they're in an fMRI scanner, or to try to look at things like sexual arousal or cooperation or empathy while a person's in an fMRI scanner. I mean, they're lying there on their backs in this very loud, noisy chamber. It's a very strange sort of situation to be recording someone in the first place. And so we have these very strange experimental paradigms that get set up that perhaps don't really tell us about how people are acting in everyday life. And then finally, there's also the crisis that's going on just in general in psychology and in neuroscience that has also had some impact on the way things are being done right now. So what do I mean by the crisis? Well, the current crisis, as at least it applies to social neuroscience, could be traced back to what's called the Voodoo paper in 2009. Now, why is it called the Voodoo paper? Well, there was a manuscript that was passed around on the internet that these authors were working on. They had like some drafts that they passed around for people to give comments on, and it was called something like Voodoo Correlations in Social Neuroscience, something like that. And then when the paper finally got accepted and published in a journal, they changed the title. And this is the paper that it had finally appeared when it appeared in print. So it was published in Perspectives on Psychological Science. And you can see the title. It was Puzzling High Correlations in fMRI Studies of Emotion, Personality, and Social Cognition. So these authors were really focusing mainly on these early fMRI studies that were being published in social neuroscience. In fact, several of the studies that I have covered in this course were covered in this article. And they were looking at these high correlations. That is, they found correlations that were much higher than you would normally expect to find in other psychology research. You could think that most psychology research would yield correlations at around 0.2 or 0.4, but they found in these fMRI studies the correlations were much higher. And so we could look at all of these different things in terms of the voxels and the behavioral measures and so on. They talk about all of this in their articles that they looked at. And then they plotted the correlations that they found from all of the things that were there. And you can see that the correlations just overall, that distribution, are pretty high, 0.5 all the way up to 1. So there were several places in these fMRI studies where you're getting very, very high correlations between certain brain regions and a psychological variable. And it just looked like amazing. Like, how is it that you could find this particular brain area was so highly correlated with something else when in the rest of psychology, we don't tend to find such high correlations? Well, what they found was that those really super high correlations are the ones that are in red, mainly. Those are called non-independent. And what that means is that these researchers, a lot of these researchers in these early years of fMRI studies, and I'm talking about between like 2000 and 2010, they would collect data And then they would do like a whole brain analysis, looking at all the different parts of the brain, and then just take all of those brain areas and correlate them with whatever the measures were that they were recording behaviorally, or the independent variable, or whatever it happened to be. And then they would find from all of those the ones that came out the best correlation. So they might find that TPJ is highly correlated and something else. Then they would rerun the analysis with using the TPJ now as the main dependent variable. And then because you've already kind of like done a whole brain analysis and figured out which areas that you think are the most activated or most correlated with the behaviors, of course, when you rerun the analyses, you get very high correlations then. And that's what they mean by non-independent. Independent would mean that you go ahead and take maybe half the data set, do a whole brain analysis, and then test the regions that seem to be most activated for your particular behavioral task or psychological manipulation, and then test it on the other half of your data. Okay, so you take half the data to figure out from the whole brain analysis which regions are most active, then test it on an independent sample. And you can see when they do that, the correlations tend to be a lot lower. And so we don't see as robust of a finding. And so that's what they meant by voodoo correlations, that they thought too much of this non-independent method was being used in the studies, and that was therefore yielding ridiculously high relationships between certain brain areas. That was all, a lot of that would just be like statistical chance, that you're getting things that wouldn't normally be there if you had done it independently, as you can see in this graph. Well, one of the major researchers that they sort of attacked in that article was Matt Lieberman, who's the editor of Social, Cognitive, and Affective Neuroscience. And Matt Lieberman at UCLA, he's the one, by the way, who was the senior author on the paper having to do with the cyberball study. Remember the cyberball study where they looked at brain activity after a participant had been excluded? Matt Lieberman's done some really fascinating research over the years, but several of these studies that had these high correlations were studies that Matt Lieberman was a co-author on, and there's Elliot Berkman and Tor Wager that you see there. And so they took it sort of offense at this voodoo paper, and even referred to it as voodoo, even though the paper itself didn't have that in the title anymore. And they argued that this was not really a problem, and they sort of went ahead and went on the defensive about it and tried to argue that there really weren't these ridiculously high correlations just because of statistical artifacts. And so they wrote a defense of this. You can go back and look at these papers. You can look at the discussions. There was even a debate at one point between Matt Lieberman and somebody else. I can't remember who it was, maybe Piotr Winkelmann, who was the co-author in the other paper, in which they were kind of going at each other at a conference and so on about this. But I think most of the stuff has been accepted now, is that we're past all of this. This is like over 10 years ago now at the time I record this lecture. And I think that now we have much better methods in terms of how we analyze fMRI data. And so that I don't think these particular correlations are really happening in a voodoo way anymore. But moving to the present, there are still a lot of controversies plaguing all of psychology. And these other kind of controversies are called questionable research practices, or QRPs. And the idea here is that just after the voodoo paper came out, is that perhaps we have a proliferation of false positives in our psychology studies. Remember, false positives are type 1 errors. These are when you reject the null hypothesis when we shouldn't. That is, we're basically saying we have a real finding there when we probably shouldn't have done that. We actually didn't have a real significant finding. There really wasn't anything going on here, but we somehow rejected the null anyway. And false positives, according to Simmons et al., have a costly error associated with them because once they appear in the literature, they are particularly persistent. They're really hard to get rid of. That is, if you went ahead and maybe reported a study where you said there was a significant finding and maybe there really wasn't a significant finding there, what actually means then is that other people will try to come along and replicate it, and they won't be able to replicate it because it wasn't a real finding to begin with. And therefore, they can't get that other paper where they have a null result reported because most journals don't like to publish papers that have null reports in them. Also, false positives waste resources. That could be whole fruitless research programs that happen or ineffective policy changes that occur based on the fact that the original study wasn't really very robust in the first place. And that also means then the field starts to lose credibility if we have lots of false positives. So how would you get a false positive? Well, in psychology, even though our nominal false positive rate is less than 5%, that's what it means when we say p less than 0.05, what Simmons et al. Argued in 2011 is that we have these researcher degrees of freedoms that increase this rate. This is where we get these questionable research practices. These are things that actually inflate the chances that we actually will say we have a finding when we really didn't have one. And so these are some of the QRPs that have been identified over the years. Selective reporting or partial publication of the data, optional stopping, p-value rounding, the file drawer effect, post hoc storytelling, and the manipulation of outliers. So all of these are research practices that would apply to all of psychology, but I'm also saying that they obviously are also going to apply here in social neuroscience. So what is selective reporting or partial publication of the data? That would mean that when you have selective reporting or publication of the data, that you're not reporting all of your findings. You may have some findings that you did in your analyses that didn't support your hypothesis. So what you do is you just delete that hypothesis from your introduction. You never mention the fact that you didn't find support for it. And it makes it look like your findings are stronger and that you were more correct than you actually were, because you're not reporting everything that didn't work out. Or you're only publishing part of the data set that supports your hypotheses and not publishing the part of the data set that doesn't. Optional stopping means that you keep collecting data until you get a significant effect. So you stop after you've run a certain number of people, you go ahead and run your analyses. Is it p less than 0.05? No, it's close, but it's not there yet. So you go ahead and run a few more people, stop, go ahead and run your analysis, and so on. The problem with this is that every time you do this, you're increasing the chance that you're going to find something just by chance. And this little graph shows this here, that the percentage of false positive results waiting after, if you just do this, like you run one more person and stop, or you wait 5 more people and stop, or 10 more people and stop. Every time you do this, you can see that you're increasing your chance of a false positive greatly. Remember, that red line is the 0.05. We want things to say that we're only going to accept false positives if they're less than 5% of the time. But if you keep doing the optional stopping after each one more person, or 5 more people, or 10 more people, or 20 more people, the real percentage of false positives is like over 15, 16%. So we're actually increasing the chances of getting false positives by doing this. So another thing would be p-value rounding. So this is a sort of a funny one to me. This means that you have 0.05 as your cutoff, and then you run your analyses. You've done your brain analyses, and you've found, like, the ACC is at 0.051. It's not 0.05. And you say, well, that's close enough. I'll just round it and say p less than 0.05. Well, 0.051 might not sound so bad to you, but what about 0.06? Is that close enough to say it's less than 0.05, or 0.065, or 0.72? As you keep increasing the number, it comes to a point where it seems to be sort of ridiculous to say you're rounding it down below 0.05. Yet people do this all the time. They actually do round the values down to say they're less than 0.05. How would you possibly know that they did this? Well, one way you could do this is to actually look at the statistics that they report in the results section, and then run an analysis that tells you what the exact p-value is. And luckily now that there's actually something out there called StatCheck, which is a program where you can take your data or a manuscript and upload it to a StatCheck. And StatCheck will actually recompute the p-values and tell you whether or not the p-values are below 0.05, and it will flag anytime the numbers don't make sense. This also, by the way, helps us with errors like, when let's say you're running your statistics and you have some value like t is equal to 1.652, and you type in the number incorrectly as you transfer it from your statistical analyses to the manuscript, this StatCheck will actually find errors like that and report them to you. Now, when they originally developed StatCheck, they went out and just tested StatCheck on a bunch of published papers, and they found many of these papers that were in some of our leading journals had these kinds of errors that showed evidence of p-value rounding. So it is a real thing. People do actually do p-value rounding, hoping that no one's going to notice that they're actually not. Then we have the file drawer effect. Now, file drawer effect isn't so malicious. This is more about the fact that our journals don't like to publish papers that don't have significant results. So you've done all this work to collect data, and then you don't find a significant effect. Like in the last lecture, you remember that Tom Denson did a study where he did self-controlled training, and he found that in his fMRI study that he got no effect of self-controlled training on the actual behavior of the participants compared to the control group, even though in previous studies they did find self-control training worked on the effects on the group. Well, let's say then he tries to submit that paper to a journal and they say, well, haha, you didn't replicate your previous effect. You didn't get a significant effect there. We can't accept your paper here. Well, instead, he was able to get that paper published, even though he didn't find that he could replicate his behavior. He did find something with his fMRI analyses. But if he hadn't published the paper at all, we wouldn't know that he wasn't able to replicate his study. And all the work and everything that he did in that would not have been there, been reported. And we might think then that every time you do self-control training, it always works. But now, because the paper's been published where he has one where it didn't work, we have a little bit more color, a little bit more understanding about what's really happening with self-control training. This is all just to go to show you that the file drawer effect is if we have a lot of papers that are out there that have never been published because they had results that didn't reach the p less than 0.05 cutoff, then we may be misleading our whole entire field about how robust effects really are, because only the ones that have positive results get affected, or get published. Then the next questionable research practice would be post hoc storytelling. It's also known as harking. Harking is hypothesizing after the results are known. This is actually a paper from 1998 by Norbert Carr, and you can see that he talked about this back in 1998. We've known about harking for quite a long time, but it wasn't until the 2010s that people really started to notice this. And the idea here is that people very often, when they are researchers collecting data, they then find that when they run their analyses, they didn't actually support their hypotheses. So what do? They go back and change the hypotheses to match their data. Now you might say, well, doesn't that make sense to do that because they were wrong to begin with? The problem is that that gives you sort of an illusion in the field that everybody's really, really smart and they already know beforehand exactly what to predict. They run the analyses and found support for their hypothesis. But if we actually knew that a person found something contrary to their hypotheses, that we might feel more cautious then about interpreting the results. So we say, well, maybe under these conditions, it doesn't really quite work. I need to know when we find support for the hypothesis, when we don't find support for the hypothesis. By changing the hypotheses so they always match the results, it's a bit harder then for us overall to understand the theory, understanding how everything really works. Manipulation of outliers is another problem. This is a problem of the fact that when you run statistical analyses on your data, that if you look at the outliers can have a profound impact on whether or not you get a significant result. And what researchers sometimes do, and I've heard people being taught this by their professors, is they'll run the analyses with the outliers included, or with the outliers excluded, or some other cutoff is used for the outliers, and they just compare and figure out which analysis works out the best. So they manipulate the outliers by running different analyses that include the outliers or not. And by doing that then, running the test 3 different ways, then finding out which one best supports their hypothesis, they're inflating their type 1 error, right? They're more likely then to falsely conclude that they have an effect when they really didn't have an effect. It was just playing around and running the analyses until they found something. Just to show you how widespread this is from the Simmons et al. Paper where they looked at reaction times, and they looked at 30 psychological science articles that referred or reported reaction times. And now you remember, a reaction time is how fast you press a button to something. And certainly some people are going to be too fast when they press the button. It's an error, like they started pressing the button before they saw the stimulus, or maybe they're too slow. That is, they, just their attention was caught with something else, and then they look back over and, oh, I better press the button. And so as a researcher who's collecting reaction times, you might decide, I got to get rid of those really fast ones or those really slow reaction times. Well, most, but not all, researchers excluded some of these responses as too fast. So in these 30 psych science articles, they had different rules, though, about what they considered to be too fast. So if you look across the different articles where they excluded things because they were too fast, some articles said that they got rid of the ones that were the fastest 2.5%. Some used a rule that faster than 2 standard deviations from the mean, or that it was faster than 100 or 150 or 200 or 300 milliseconds. So right there, how can reaction time studies have such variation in what they call something to be too fast? And then look at what happened with too slow. Too slow responses in these articles were defined as either like the slowest 2.5%, or they're the slowest 10%, or that they were 2 or 2.5 or 3 standard deviations slower than the mean, or they were 1.5 standard deviations slower than the conditions mean, or they were slower than 1,000 or 1,200 or 1,500 or 2,000 or 3,000 or 5,000 milliseconds. Why am I bringing all that up? I'm trying to show you that What that suggests is that among these 30 psych science articles, that the researchers may have been playing around with what their cutoffs were for their outliers. They're manipulating their outliers and running different analyses until they found the analysis that gave them support for their hypothesis. Because how else can you explain such a wide variety of cutoffs for what's too fast or too slow? You might expect maybe just a couple rules that everybody would use, but people are actually using quite different rules, which suggests that they're maybe modeling different outcomes here until they get a significant result. So again, inflating our type 1 errors. So I've covered a bunch of these questionable research practices. All of them could be things, again, that apply to social neuroscience. And the cumulative effect of all of these questionable research practices is that our published findings then are probably much stronger than they actually are. That is, the published data, or the way that we talk about the results, make it seem like we have really robust, strong effects, but they're not actually as strong as they seem to be, right? And what we can see this is a very important paper that was published in Science in August of 2015 by a group of psychology researchers led by Brian Nosek at the University of Virginia. And what they did is they took 100 studies that have been published in recent top journals in psychology and tried to replicate them. So they went ahead and asked researchers around the world to help them replicate each of these studies that they looked at, these 100 different studies. They tried to contact the authors of the original studies to make sure that they were following their methods as best they could, using very similar samples, whatever it would take. They tried to work with those people when they could. And then they looked at whether or not they could replicate exactly the same findings when they ran the study just a few years after the original study had been done. And what they found was that only 39% of the replicated studies had significant results. So when they looked at 39 out of 100 studies, 39 of them showed significant results that were replicable, that they found the same thing. You can see here sort of a graph. It shows you what the original effect size of the study was and what they found in terms of whether or not they could replicate it. And basically, the stronger the original effect was, the more likely it was that they could replicate the study. But there were a lot of studies that they couldn't replicate, and they had very small effect sizes. In fact, they looked at a lot of different studies here, and you can see there's all these different characteristics that they analyzed. But basically, it comes down to the fact that a large number of these studies just couldn't be replicated, suggesting that maybe there's a lot of false positives there in these top psychology journals. It caused a real crisis in our field. There were several people who, by the way, took issue with the 2015 Science paper, saying that it was full of statistical errors, other methodological problems. I have to say that a lot of senior people in our field reacted quite negatively to all of this because they felt like it was sort of an attack on their standing in the field, that these young upstarts come along and try to replicate their studies and they do a bad job of it and then try to bring everybody down because of it. A lot of hand-wringing has been going on over the last 7 years. I do feel that now, at the time that I record this lecture, that we've now moved to a more mature, positive sort of response to all of this, where you're seeing a lot of positive changes happening in our field to reduce the chances of false positives and therefore maybe increasing the replicability of our findings. But I think the jury's still out in seeing what happens in the long run. I'll mention, by the way, that all of this stuff that I just talked about was just, none of that had to do with neuroscience, in the sense that those were all leading psychology journals that didn't actually have any neuroscience findings in it. But people have also been making these same sort of judgments and same sort of criticisms of neuroscience. Here's a paper, by the way, published in 2013, looking at how small sample sizes undermine the reliability of neuroscience. That is, the problem is that, according to Button and her colleagues, was that in neuroscience, they very often run with very small samples. And you might have noticed I've highlighted in a lot of lectures this semester, especially studies that were done between 2000 and 2010, how small the sample sizes were. Think about, for instance, some of those studies on morality that were published with just fewer than 20 people in the study. Very small samples that were done and got published in Nature or Science and had huge impacts on our field with small sample sizes. Well, it tends to be that when you have small sample sizes, it's harder to replicate the study. We don't have very much power there. And in fact, in this paper by Button et al., they looked at power across different neuroscience studies and found, if you think of 80% power as our usual cutoff, we usually like to have a study have about 80% power to be able to detect a real finding, okay? At 80% power, you can see that most of these studies are below that. Most neuroscience studies have much smaller sample sizes, have a lot less power, and they're probably therefore not going to detect a significant effect unless they use some QRPs. They do some massaging of their data. They do voodoo correlations, whatever it's going to take to get a significant result because they're using such small sample sizes. And so what Button et al. Argued in 2013 was that neuroscientists needed to increase their sample sizes. It costs a lot to do this. I can tell you that if I want to go ahead and run a study with 120 people, it's going to cost me tens of thousands of dollars to do such a study in an fMRI study. I don't have that kind of money, so maybe I only get $10,000. I'm going to run a much smaller fMRI study as a result. Well, what could you do about that? They mentioned some things like, first of all, you should be doing a priori power calculations before you run your study to figure out how underpowered it actually is. They should be disclosing the methods and findings transparently. They should try to pre-register their study protocol and their analysis plan, make the study materials and the data available. And finally, look at the bottom, work collaboratively to increase power and replicate findings. So what they suggest is if it's really that hard for you because you don't have that much money to pay for your fMRI scans, maybe combine forces with researchers at other universities together you could collect your samples together, or collect your hormone analyses that way, or genetics, or whatever it happens to be, because you could work together collaboratively across different labs around the world to increase the sample sizes, and then that study will have increased power and be more likely than we could replicate the findings. Even this paper here published in 2016 suggesting that intranasal oxytocin studies are also probably underpowered. This was Wallam et al. In 2016, and you can see there they say in yellow, our conclusion is that intranasal OT studies, so these are studies where they go ahead and apply oxytocin through the nose, are generally underpowered, and that there is a high probability that most of the published intranasal OT findings do not represent true effects. So again, because they're underpowered, you might say, well, how do they get significant results? They probably get significant results through some of those QRPs. They probably do things like deal with outliers, do post hoc hypothesis changing, rounding of the p-values, whatever it takes, so they can actually basically p-hack, get it down below 0.05 because they don't have enough power to begin with. So it's actually quite rare because it costs so much money to intranasal oxytocin to have very large sample sizes. I was involved in some research headed up by Julie Henry and Sarah Granger at the University of Queensland, where we did some intranasal oxytocin studies a few years ago. And one of the things that we were able to do is actually pay for much higher sample sizes so we could look at oxytocin's effects on how people perceived emotions and others. And we found that some of the findings that others had reported we couldn't replicate when we had a much bigger sample size. And so I do think that a lot of these oxytocin effects could be not real in the sense that people can't really replicate them, and they might have been false positives to begin with. So what do we do to kind of reduce all of this? How do we move forward to make things better? And there's been many solutions offered to this, and we could look at this both at the researcher level and at the level of more like the science and the society. One thing obviously you could do that I keep harping on about here is increasing the sample size. So instead of being satisfied with a small sample just because it's really expensive, try to do what you can do to increase the sample size, like 60 to 75 participants per condition at a minimum would be good to have. Report all your measures, your conditions, your procedures that you use in the study. Be transparent. Don't hide things that didn't work out. You could include this 21-word statement in your article where you say things like, we report how we determined our sample size, all data exclusions, if any, all manipulations, and all measures in the study, so that we can go ahead and say to other people, we're not hiding anything from you. We actually are telling you exactly what we did in this study. Do not use post hoc explanations as original predictions, so basically avoid harking. And finally, you could pre-register your study by going to the Open Science Framework or openscience.io. And what you do there is before you actually collect your data, you register it there. You say, what are your hypotheses? How are you going to collect the data? Everything you did before you actually go in and run your analyses so that you can show that you were being transparent right from the start. What about at the journal or the field level. Well, what we could do is we could welcome pre-registered studies, particularly those that are aimed to replicate prior research. So one thing that's been happening is a lot of journals now have little badges that they put on the article that show that the article that this researcher actually did pre-registration, or that they shared their data or whatever. You could also record that authors report all of their measures, conditions, procedures used in the study by making them sign a statement that says that. They should also welcome exploratory and confirmatory research. You see, one of the problems is that's probably happened in our field is that a lot of us just want to have confirmation research where we have a hypothesis that gets tested, and then we can say whether or not we supported the hypothesis. But it's also important to do exploratory research where we don't really know beforehand what we're going to find in this new paradigm in brain activity, for example. So we go ahead and do something that's more exploratory, and because it's not confirmatory, it's been hard to publish those kinds of papers. So if the journals open themselves up to allowing more exploratory research to occur, then we might be able to get away from a lot of that harking and other post hoc reasoning that happens. And try to educate our students about best practices, because the students are actually the future. You watching this lecture are the future of social neuroscience if you're going to go into our field, and having you know about this kind of stuff, about open science and being transparent and reducing QRPs is really good. In fact, we've been trying to do this at UQ for a few years now. About 4 years ago, I organized a conference that we had at UQ on open science, and these are some students that were actually in my social neuroscience course who helped out with our open science conference that we had at UQ. So we're trying to get more people educated about this. It continues to happen, and I think that there are a lot of people who are open to this, and we're going to see positive changes keep occurring in our field. All right. On that sort of positive note, let's move on to the second part of this lecture, which is I want to talk about the next wave of social neuroscience. Some important developments that I think are going to pave the way to social neuroscience in the next 10 years. One thing I just want to highlight, this is something I referred to in an earlier lecture, is just greater attention to neurosexism and actually neuroethics generally in our field. I mentioned to you that Cordelia Fine has published a couple books about neurosexism, like Delusions of Gender, that was published in 2010, also Testosterone Rex in 2017. Both of these books, she argues that a lot of people who are doing research in neuroscience and social neuroscience have sort of are using the neuroscience to justify inequities between the sexes. And so when you think about gender and sex and possibly what it means to find differences in the brain or in hormones or whatever it happens to be, Cordelia Fine says that a lot of these biases that we may already have in culture then get applied to the way the study gets designed and the way we interpret the data. So if you ever do have the time, I'd recommend that you pick up one or both of these books and read them. They're very fascinating and I think are important things to pay attention to. Neuroethics, by the way, is another important development that's been happening in recent years. This is sort of looking at some of the issues that we've actually brought up in workshops and in lecture in terms of lots of different new things that are popping up about, now that we know more about what underlies our social behaviors, what does it mean for ethics, and how do we pay attention to ethical issues that might result from things like brain injuries and other things. So you can see here's a issue that was published in 2015 in this journal called Neuroethics, and it was just about concussions. And they're looking at ethical issues around concussions, so ethical issues surrounding concussions in player safety in professional ice hockey, sport-related neurotrauma and neuroprotection, are return-to-play protocols justified by paternalism, and on treating athletes with banned substances, the relationship between mild traumatic brain injury, hypopituitarism, and hormone replacement theory. So it's just trying to say to you, the more we learn about how all of these systems interact and affect our social behaviors, and then the social behaviors themselves have tons of ramifications about how we interact with each other in society, that's where ethics can come into play here. And so this is a field that's quickly growing, just focused on these ethical issues around neuroscience and social neuroscience. All right, that's one development, neurosexism and neuroethics. Another thing I think is really important that's taken a while to catch on, but I'm seeing it more and more now, is this whole idea of hyperscanning. This involves the simultaneous scanning of fMRIs during linked social interactions. The whole point of this is something that, by the way, that Reed Montague, who we talked about earlier in the semester. Reed Montague wrote a paper about this back in 2002, but it's just now finally, I'm seeing it, like I said, more often. The point here is that we could, instead of having just one person alone in an fMRI scanner and call that social neuroscience, they're all by themselves and we present stimuli to them. How about we have two people, each of them in an fMRI scanner, that interact somehow while we go ahead and scan their brains? And so the suggestion here is that we now have fast internet speeds, we have lots of things going on where we could actually have people in two different scanners, maybe one in Australia, another one in Great Britain, and the two people are interacting through computer connections here, and they're both being scanned at the same time, so we can see how their brains affect one another. Now that's kind of still pretty expensive because it does involve then two fMRI scanners. Imagine if you wanted to do a group of 6 people and you had them each in 6 different scanners. Lots of scanning, lots of costs there. So another sort of solution besides doing fMRI simultaneously would be to do other kinds of like EEG recording. And here's something called functional near-infrared spectroscopy, or FNIRS. This is another newer method of looking at brain activity. What you do here is you actually send little light sources in. You can see there in the lower left corner. You send some light through the skull, and then you see how it reflects back up. And based on what's going on in terms of brain activity on the bottom, underneath there, you could reflect the light up differently because of the blood flow that's going on. And so because of the blood flow activity and the way the lights get reflected back up, you can get an idea of cortical activation along the surface there at different spots. And so in one case, by the way, this FNIRS has been around for quite a while now, and it's a popular newer method that you find in psychology and in social neuroscience. You can actually do hyperscanning with this. You can actually connect people in two different caps where they're actually doing FNIRS on it. And here's an example of this. This is something I found on the internet a while ago where they're doing hyperscanning while people are playing music together. So for instance, if you were interested in how two musicians like listen to each other and influence each other while they're playing, you could use hyperscanning to do this. So here's an example of this. Now, to tell you the truth, I don't really know what we saw there in terms of brain activity. I mean, obviously you could see like there seemed to be some sort of influences going back there, but in that example from the video, I don't really know what they were extracting from that to understand anything about how the two musicians are influencing each other. I'm just showing you that there's technology there that maybe under the right conditions, we could actually find some really interesting science that could happen from doing hyperscanning this way. But as I said, I couldn't find any research here yet that was reported that would use those particular methods. Now, moving on to our third sort of new development, these would be just new tools for aggregation and analysis. And Tor Wager here, by the way, he was one of those people on that Matt Lieberman paper that responded to the voodoo correlations paper. Tor has really been doing a lot to advance open science and fMRI and neuroimaging and sharing people's data and tools. And so, for instance, you can go to his laboratory page and you can see he's got a whole bunch of different kinds of toolboxes that can be used to look at brain activity in terms of how we can do analyses. There's maps up there. You can look at summaries of findings in terms of what brain regions have been found for different kinds of different manipulations. The whole point of this is that instead of just like keeping everything in different research articles, we could have like a common bunch of toolboxes, a toolbox full of tools and pictures and data that could be shared across different labs in a more productive way. And so you can look at him as possibly being a leader in that area. Another interesting development this way is the Human Connectome Project. This was a project that began in 2009. I think it involved originally funding from 4 different US universities and laboratories. And what they were trying to do here is they're looking at the connections between different brain areas and trying to get a map of how across many, many adult brains, across a whole bunch of different labs and tasks, how each of the different brain regions are connected to one another. And so this was meant to be like a roadmap that shows you what all the connections are. And it went on for, like I said, 5 years, I think, but it still officially hasn't ended. And so you can still find it on there, and there are still people contributing to this Human Connectome Project that supposedly will be just about as important as, for instance, the Genome Project was in terms of mapping human DNA. All right, our fourth development for the future is pursuing the subcortex. Now, it turns out that the subcortex makes up 25% of the human brain, yet only 7% of this part of the brain has been represented in standard MRI atlases due to the difficulties in imaging these particular regions. If you think back about it, this semester in these lectures, I pretty much if we've been talking about brain activity, we've been talking about stuff in the cortex. But the human subcortex could also obviously have important ramifications for social behavior. And so in order to access those, we haven't been able to really do it very well because of the problems of doing brain imaging and getting past the cortex. But now there are new techniques that are available to investigate the subcortex. And this paper that was published in 2017 in Nature Reviews Neuroscience mentions and reviews some of these new techniques, new things you can do with fMRI and other kinds of methods to analyze your data, to start looking at the subcortex and seeing its role. So I think that's another important development. You're going to see more attention given to the subcortex as we get beyond just imaging the cortical regions. And finally, the fifth thing I can tell you about is this interesting talk by Polina Anikieva. It's a TED talk, and it's called Rethinking the Brain-Machine Interface. And what she's talking about here, and there's other people who have been looking at this, is what we could do to better integrate AI, artificial intelligence, robotics, that kind of stuff, with brain activity. And so you can get the two of them to more directly link to each other through lots of different kinds of devices, whether it's through EEG or implanted electrodes or whatever. And she reviews this. And so I think brain AI interfaces would be another important thing. And so if you're interested in this, I recommend that you go and find her TEDx talk and watch it and see what she has to say about this. This is actually a really cool TED talk. All right, let's go to part 3 of today's lecture, which is, so you want to be a social neuroscientist. So I wanted to end this entire series of lectures by just addressing anybody who is out there who may be feeling kind of excited about social neuroscience, wants to do this kind of work like Sapolsky or me or other people. What do you have to do in order to become a social neuroscientist? Well, obviously, you're going to have to do more work at research, more training at university. You're going to have to do more research and so on. And so just to kind of give you a sense of this, and this is largely, I'm writing this from the perspective of somebody in an Australian university, but it largely applies to any place else in the world, is that there are things you can do with a bachelor's degree as an undergraduate degree in psychology or neuroscience. One of the paths is that after you've gotten your bachelor's degree, you could go off and do a PhD or an MPhil. So you could do like a master's degree or a PhD, and that would be your next step. But that would require that you get a bachelor's degree first. So you have to do that as an undergraduate. Another thing you could do is you could become a research assistant with a bachelor's degree. So you could end up working in a social neuroscience lab as a research assistant, collecting data, being the experimenter, hooking up electrodes, that kind of stuff. Or there might be other professional staff jobs that you could get as like somebody who organizes the research, makes the phone calls, does a lot of the bureaucracy, administrative stuff that could also help out with the lab. All of those are possible pathways for you to do more research, at least at the university level, that would require the bachelor's degree. But if you want to go beyond that, you're going to have to get a PhD. And PhD is required, for instance, if you want to work at a university as a professor, associate professor, senior lecturer, or lecturer. In Australia, that is actually our sort of hierarchy here. You start off as a lecturer, then get promoted to senior lecturer, then get promoted to associate, and then get promoted to professor. In the US, for example, they don't have that a senior lecturer, lecturer level. They just call them assistant professors. Then they get promoted to associate or full professors. But how do you become a professor? Well, obviously you have to have a PhD, but these days, most people, after they've received their PhD, would then go off and do what's called a postdoc or become a research fellow. So that would mean from anywhere from a year to 5, 6, 10 years, you may have to continue to work in someone else's laboratory as a postdoctoral fellow. That is, you've got your PhD, you're working more than a research assistant, like you could actually be doing your own research, but you're still doing it under the framework of another professor's lab. And that's kind of a common pathway these days, is that you go and do that for a few years. You build up your curriculum vitae, your resume, so you have more publications, more grants, things that you've done like that. And then you apply to become a lecturer, senior lecturer. Now, some people do go directly from PhD to lecturer, but I'd say that in neuroscience, especially in social neuroscience, you're going to have to probably go off to that other level there and go to postdocs or research fellows. So you could be talking about, it's not until your early 30s, if you just go straight from high school at the age of 18 all the way through all of this education, postdocs and stuff, it could be your early 30s before you actually land a position at a university as a lecturer or senior lecturer. That's just sort of my basic take on it, and that's where most of this research in social neuroscience would have been done, is in this sort of pathway. Now, when you're thinking about your future in neuroscience, one thing you could do is you could look at this interesting article that was published in 2008 in the journal Nature Reviews Neuroscience. And what they did was they asked questions of top neuroscientists at the time, Bart Fye, Insel, Fischel, Rothwell. Most of these people that are doing neuroscience in this article are wet lab, more micro-oriented. So what a wet lab means is that they're not necessarily studying humans, they're not doing brain imaging per se. They're probably working with fish or rats or other animals or prairie voles or whatever it is. And so there's like surgery involved, and they might be doing staining techniques and things like that. That's the kind of neuroscientists that they interviewed here. But I think a lot of their advice that they give about going into neuroscience applies equally to people who are going into social neuroscience. And so anyway, this is an interesting article that kind of gives you some advice about it. And what factors should you consider when choosing the field of study, they said. So they're saying the question they're asking these neuroscientists is, how do you know what you should study in social neuroscience or in neuroscience? Well, they say ignore the hot topics as they come and go. So for example, if there's some new hot topic, like let's say mirror neurons are all the rage, try to ignore something like mirror neurons, because as we saw, for instance, with mirror neurons, the interest in mirror neurons has greatly waned. It was sort of a hot topic for a few years, and then it went away. So instead, it'd be better to find something that is a problem that people have been working on quite a while, and it's probably going to be the case that there's not going to be any easy solutions to it, and people will keep working on it. All right, so pick something that kind of has some more longevity to it. Try to avoid the hot topics. They also say that you should try to find a great mentor who's made a significant contribution to science, who maybe has some name recognition, and whose laboratory attracts the brightest young people. They thought this was the most important thing. Now, this kind of makes sense from their perspective, because they're all noted people in their fields, and they're saying that basically success begats success. So you as an up-and-coming person, the more you can find a successful group, the more likely you're going to benefit from their success as well. Of course, one thing to kind of think about for that is that perhaps you'll just be like a cog in the machinery of that particular lab, that, it's going to be hard for you to shine if everybody else is working for the great successes of the leader of the lab, the top person in that field, in that lab. And so you're going to be hard to maybe come and shine. But these particular scientists didn't think that was a problem. But I do urge you to feel some caution about this, that you could get too close to such a shining star that they throw such a shadow on you that no one's actually going to notice you, because That people are always going to say, well, yeah, the only success that came to that person is because they were working in famous person X's lab, right? I would say the more important piece of advice that they bring up here is to be truly excited about what you're doing. That increases your chances of success, because going into a field like neuroscience is really hard to keep your motivation levels up all the time. There's lots of places where there's going to be failure coming along, where you run a study, it doesn't work out after you put in hours and hours and hours and days and maybe years on the study, and it doesn't yield anything of any consequence. Or maybe you've applied for a grant, didn't get funded, or you submit your papers. So you're going to have to find other things besides the external rewards to keep you motivated. So having that intrinsic motivation, because you're really interested in this topic and that really excites you, is going to increase your chance of success because you're going to stay motivated. And so they say, work where discoveries are being made. Try to go someplace where there's a lot of excitement going on. Again, balancing the idea you want to stay away from hot topics, but work where the discoveries are being made. Choose an important question, one that addresses a fundamental issue in the field. Trying to pick some little topic that hardly anybody's interested in is probably not a good idea, okay? So pick something that's going to be a fundamental topic. And I think any of the topics that I've covered in this course, in any of these lectures, you could see would be good topics to get into. So what is the best way for a neuroscientist early in their career to establish contacts with other neuroscientists? And again, this comes from that article from Nature Reviews Neuroscience. They suggest these top neuroscientists to attend as many research meetings as possible, preferably the smaller conferences, 100 to 300 people. So what they're suggesting here is that we have many such research meetings every year. And the problem, of course, is when you live in a country that's far away from everybody else, like those of us who live in Australia, it can be very expensive to attend research meetings. So we do try to go to research meetings that are nearby. So for example, I'm going to be soon attending a conference that's in Sydney, which is only like an hour flight from where I am. But going to someplace in Europe or the United States is going to cost me a lot more money. And I'm going to have to be really careful about what research meetings I'm going to go to as somebody who lives in Australia. But the general point here is to try to go to as many of these as you can, because you can then find out what's going on. People are talking about the newest research findings. It gives you a chance to actually talk to the researchers. A lot of the people that I've mentioned in the lectures in this course are people that I actually met only from going to research conferences. And the reason why they emphasize smaller conferences is that there are some very, very large conferences, like the Society for Neuroscience, I think has somewhere between 10 and 20,000 people that go to that conference. It's really hard to network where there's 10 to 20,000 people. Trying to find the author of that one paper that you're really interested in, bumping into them at coffee is going to be hard when there's 10,000 people there. But if you go to a smaller conference that's really kind of focused on your topic, like let's say you're interested in anger-provoked aggression, and there's a small conference just on that, or there's a conference on empathy or sexual attraction. Going to a small conference like that, and there's only 100 to 300 people there, you're much more likely to meet other people who are also interested in your topic and have dinner with them, chat with them, learn more about their research, and possibly even collaborate with them on some new projects. You could also, they say, get involved in organizing a seminar series or a conference, because if you organize your own conference, you organize a seminar series, you're going to get to know the people who are in that field as you organize the speakers and bring them in. The funny thing was that in 2008, when they wrote that article, they weren't too sure about the internet yet. That is, they don't really have good advice about how to use the internet for social networking. But I think you know from COVID times and other things now from social media that obviously the social media becomes really important for networking professionally in all sorts of different domains, even in neuroscience. And so one thing I strongly recommend that you would do is to get onto Twitter. Twitter has a whole section of academics who are interested in using Twitter to promote research, to talk about research topics. A lot of the stuff I've talked about in this lecture about open science was first talked about on Twitter. There's a large, thriving group of people interested in social neuroscience that I follow on Twitter. So getting on Twitter and following some people there, even if you never make a response, you don't post anything yourself, you can learn about some new research, find out about how people think about things, where they might be at a conference, that kind of stuff. So I do recommend using the internet much more than they would have talked about in 2008. All right. So that is my last lecture on social neuroscience. I hope you've enjoyed this. We did talk about general limitations and strengths of social neuroscience today. We talked about some developments that I think are coming in the next wave of social neuroscience. And finally, I've given you some advice that came from that, largely from that 2008 article about neuroscientists. But that's all I have. I do encourage you that if you're interested in social neuroscience, you get in contact with me in the future. I'm on Twitter. You could follow me. I'm Social Neuro there on Twitter. And I also have a Wet Lab webpage. And that's all I have for now. Thank you very much.